Thank you, John, and thank you, everybody, for being here today. I know it's not always easy to make the trip and visit us like this, but we really appreciate it. And those of you taking the time to watch from home or from your offices, thank you for taking that time as well. I always want to open the summit by just reminding everybody that we're talking to a very broad audience, right? So we are presenting science about ALS and research here, but we're speaking to people who, in some cases, don't have a really deep background in biology and drug discovery. And we're also talking to folks who are ALS experts, clinicians, and researchers who do have that deep background. So everybody's going to end up being probably a little bit uncomfortable, right? So if you're a person with a lot of background in ALS, we might not be sharing as much detailed science as you're used to seeing in platform presentations. And if you're a person who doesn't have as much background, we're probably going to say some words that you don't know. We'll try to explain them as we go, but if there's something that comes up that's not familiar, feel free to find us. You can find me, find the rest of the science team members from TDI here, and ask us questions throughout the day. We're here to tell you more about ALS research. So today I'm going to be focusing on collaborative science in the service of ending ALS. That's going to be a theme for the whole day, because that was a theme for ALS TDI this year. We've always been a collaborative institution, but we've been really thoughtful about how to be very explicitly open about the data that we've collected and share it with the community, because we want to make the broadest impact that we possibly can. And we know that we can do that better together. So I'm starting with this figure. If you've been to summits in the past couple of years, or if you've attended a couple of our town halls, the figure might be familiar to you. It's a representation of the steps that are often carried out to arrive at a medicine. So a lot of medicines that are life-changing in the world that we live in today have gone through these processes to be arrived at. We have a dearth of those in ALS still, but it is likely that a lot of medicines that are developed will go through this process. So if you're not familiar, I'm going to walk through some of these steps pretty quickly and tell you more about how we apply these steps in collaborations. So the first step that I want to talk about is fundamental research, or basic research. This is often the step that is carried out, usually in academic research labs, where it's science for the sake of science sake. You're not really, there's no specific objective other than learning more about the world. And this is important in the context of a disease like ALS, because we still have a lot to find out. There's still a lot to learn. And what often comes out of fundamental research are targets. So targets are biological intervention points that you can imagine applying or developing a medicine for that will go and change it, make it go up, make it go down, and have good consequences biologically to ameliorate a disease. Once you've identified a therapeutic target, you often move into a stage called drug screening. So you build systems, often in a lab, that give you the opportunity to test many, many ideas, or often molecules, drugs, that might modulate that target. You're looking for things that'll hit the target. Most things don't hit the target, but things that do are very creatively called hits. So you're trying to identify hits. Once you've sifted through many, many ideas and identified these hits, you then need to understand more about how those hits could work to identify something that has a chance to be a real medicine. And in order to do that, you need to test them in more complex systems than you might have set up in that initial screen, which is usually cells in a dish or proteins in a tube, and you're looking at their activity. Now you're looking at a complex system which might be an animal. And you're usually using animal models that represent the disease that you're trying to study. So for us, we often use mouse models that represent ALS or different types of ALS. We also use zebrafish as well. And you go through this process to find molecules that are efficacious. They make those animals live a better and longer life while also not doing harm. So they're not going to cause problems in the liver or the kidneys or the blood compartment, things like that. So you're looking for what can be a lead molecule. That's your lead. Once you've identified a lead molecule, something that has potential to be a drug, you need to advance it into these studies that are called IND-enabling studies. So IND means investigational new drug. An investigational new drug, that's a designation from a regulatory agency like the FDA or like the EMU in Europe or equivalent agency in Japan, et cetera. These are studies that often don't have anything directly to do with the disease that you're studying. They have everything to do with the blocking and tackling of knowing exactly what your drug does, how it behaves. So you're figuring out the manufacturing. It is the responsibility of the organization that's developing the drug to produce a product that'll behave itself over and over every time you make it. So batches should behave the same way. And it's also the responsibility of the organization to understand the upper limits of how you can use the medicine. So what are the toxicity profiles? What are the types of things that a clinician would need to worry about if they were actually going to deliver this to a person in a trial? Once you've completed these IND-enabling studies, you then have your drug product, right? That's the actual entity that you're allowed to move into human clinical trials. You can test in people, and those are your clinical studies. Clinical studies are often referred to as like a phase 1, a phase 2, a phase 3 clinical trial. And these are the studies where you're now taking your new medicine, testing it in people, first looking and primarily for safety. You want to see that what you learned in those IND-enabling studies is bearing out now that you're really in people. You understand the safety profile. And then you're in the phase 2 and phase 3, you're looking for efficacy. You're looking to see if that medicine is actually helping ameliorate the disease. And so that's what you're getting here, that your product is safety and efficacy. And once you've achieved safety and efficacy, then you have an approved medicine. And that's what we're striving for. And that's what your team at ALS TDI is working on every day, are working on different steps of this process to get to the point where we have more approved medicines for ALS. We try to do that as quickly as we possibly can. We know that ALS is devastating and aggressive, and it impacts people far too quickly, and we can never move fast enough. A couple years ago, I stood up here and I said that we had an objective to try to deliver a medicine every 2 years into human clinical development. We haven't met that objective. It's a really ambitious objective. That's not to say that we haven't made progress. We've made progress on multiple programs. We have a set of drugs that we've talked about in the past that you could hear more about if you ask us about it. Copper complexes. We have a program going after a series of enzymes called PRMTs. We have PRMT inhibitors. And we've made progress, but we don't have a medicine ready for the clinic today. That said, I want to maintain that aggressive stance. I want to maintain that standard. I want to push for those objectives every 2 years, because every 2 years is the longest possible time that we can tolerate for a person who's diagnosed with ALS today. So we're going to keep pushing for that, and we'll always strive to achieve that. But we do need to find ways to accelerate. Now, when I've presented this in the past, I framed it this way. I've said that the work that happens in ALS TDI, within the four walls of our lab at TDI, we do fundamental research, we do drug screening, and we do lead optimization, and then we work with partners on the IND-enabling steps in the clinical studies. That was actually an oversimplification. That's largely true, but it's more complex than that. We actually do have clinical studies that happen at ALS TDI. They're not interventional studies. We're not testing drugs in people with ALS, but we are learning directly from people with ALS through the ARC study that John alluded to earlier. And we work with partners across the entire gamut. There's no step in this process where we are working alone. And that's actually what I want to focus on with the rest of my presentation today. So the first step, fundamental research. So remember, this is the step where you're looking for targets. We often work with other investigators, sometimes in academic spaces, sometimes in industry spaces, where we have non-overlapping expertise, right? We have a lot of focused expertise on ALS biology, but there are these subdomains where we might not have developed expertise. And one example that you'll hear about today from Theo Hatzapetros when he talks later this morning is a collaboration with a group at Brown, Gregorio Valdez. I've known Greg for, maybe 15 years now, and Greg is an expert in neuromuscular junction biology. And he spent 20 years studying that specific domain. So we have motor neuron expertise, we have animal model expertise, but this guy knows everything there is to know about NMJs. And so we work with him in that domain. That's an example of where we have non-overlapping expertise, and we hope that sharing data and sharing models and sharing experiments gives us a position to arrive at better therapeutic targets. In the context of drug screening, our team, you'll hear from Kyle Denton later today about a lot of our cell-based assays and our induced pluripotent stem cells. We build a lot of systems that allow us to screen a lot of drugs, but we're not the only ones that have drugs to bring to those systems. A lot of folks have ideas on things that could be tested. And so we collaborate to test new drugs in those cell-based assays. And you'll hear from Dr. Jim Larrick this afternoon about these types of collaborations that we have. We actually have a partnership with Corsilex, where Jim Larrick works. In the context of lead selection, testing drugs in animal models of ALS, this is something we've been doing for a really, really long time. We probably have as much, if not more, expertise in this domain than anybody in the world. And so a lot of companies and academic institutions come to us to do the pharmacology, right? So testing how drugs behave in the animal models to identify if they, in fact, have a lead molecule that they can move into the clinic. One example of a partnership like this was a project with a company called Neuroimmune. Neuroimmune had a number of monoclonal antibodies, so they were a type of drug that targets misfolded SOD1, and they asked us to evaluate many of those molecules in our animal models to help them identify which one might be the best one to move into the clinic and at what doses and concentrations they would want to do that. They did end up selecting a molecule that became known as AP101, which is currently in a phase 2 clinical trial today. And you'll actually be hearing from Theo Hatzapetros as well about probably, I think he's going to talk about 4 different programs like this that are collaborative, where companies or academic labs have drug ideas that we're testing in our lab today. IND-enabling studies, I'm not going to belabor this. These are not specific to ALS. These are studies that you often have to do without any necessary expertise in ALS, and there are a lot of companies that are really, really good at this. So we go straight to those companies like Charles River Laboratories or Lonza Therapeutics, things like that, to help execute these studies. But I do want to talk a little bit about the clinical studies that we execute under the auspices of the ALS Research Collaborative, or ARC study. So the ARC study is a collaboration in a few different ways. You're going to hear a lot more about this from Alan in the next presentation. But this is a collaboration with people with ALS, first and foremost. People with ALS are sharing their data with us. They're teaching us because they are experts in their disease. And so they're teaching us about their disease by generously sharing data and sharing samples, blood samples, et cetera. And we treat them as partners in that we want to share the data back. And we want to make sure that they have access to the information that we generate through this study. But we also kind of took a next step in making this a much broader collaboration by the establishment of the ARC Data Commons this past year. The ARC Data Commons puts all of the data that we've collected from nearly 1,000 people with ALS over the past 8 or so years and puts it all on the cloud, all on Google Cloud. And we set up a platform using a Google tool called Looker, which allows for the easy download and easy analysis of the data that we've collected. And we share those data with essentially anyone who's a stakeholder in the ALS space. So we're sharing those data with academic researchers, we're sharing those data with other nonprofits within the ALS space, but also folks who are outside of the ALS space, because we want to bring more people into the solution. And so we're sharing those data, and we also share those data with industry, companies that are working to develop new therapeutic targets or new drugs of their own right. And so I'm really proud of the progress that we've made in opening these data up. All of that feeds back into new targets, new drugs, the whole new process. So we do work across the entire ALS drug discovery pipeline. We do it in a lot of different ways. We have our own mission, right? We want to invent our own drugs, and we want to advance them through the pipeline as quickly as we possibly can. But we also work in a way that is aiming to accelerate the work of others who have a shared mission of also ending ALS. We don't actually care where the treatment comes from. We just care that it happens. We don't care who, we care when, right? So we're trying to accelerate that. And I was thinking about this over the past few weeks, and more recently I've been thinking, I don't know why, I'm a dad, and so I've been thinking more about my dad. We didn't always see eye to eye, but now I kind of get him more now. My dad, you do, don't you? My dad was a Navy sea captain. He was a sea captain in an outfit in the Navy called Military Sea Lift Command. And the mission of Military Sea Lift Command is, yes, they have their own specific missions, each vessel. They have to make deliveries to Navy seaports around the world so that those seaports can execute on their missions. And they also end up, along the way, having to undergo activities or missions like these, little side quests, I guess you would say, called underway replenishment. And so what you're looking at here is I have a picture like this on my wall. The middle vessel is the one that my dad would have captained. And they actually, while they're executing their own mission, they do underway replenishment where they're sharing resources, fuel, food, medical supplies, and munitions to others who have the broader mission of protecting the nation, right? And so they accelerate and support the mission of everybody else in the service to the greater mission. And I like to think of us a little bit that way. We have our own work, we have our own projects, and we are steaming forward to get that job done. But it's our job to help other people move faster too, because we'll get there faster that way. With that, I just want to show just a few of the different groups and organizations that we've collaborated with over the past few years. I'd be remiss to not mention specifically Augie's Quest, who have always been our stalwart supporters and have fueled the work that we do to keep our engine moving forward. But really, so many different groups here are committed to ending ALS. And with that, if we have time, I'd love to take questions.
Good morning. I'm Meg Reyes, and my husband's Juan Reyes. And so one of the questions I have for you is, how many do you traditionally have in your lab through clinical, or like getting up to that IND process, or that little bracket that you had that's within your four walls primarily? How many
Yeah.
do you normally work throughout a time?
So it depends on how you think about it. So if you're talking about the drug screening stage, we could so that second bucket, we could be looking at thousands in a given year, right? Screening through thousands. If you're talking about the lead optimization, lead selection stage, so when you're testing in animals, we're probably looking at about 20 per year. Most things don't work, right? ALS is hard. But we're trying not to fool ourselves. We want to only advance the things that make the most sense, where the data justify it. But yeah, we've got a team that's working pretty hard all the time. Please.
So on that note, when we talk to people with ALS and we talk about potential cures or potential, like, keeping it chronic, taking a medication that allow you to continue with ALS, living chronically at a potentially paused stage, one of the things that I always kind of feel like, because ALS, I'm not going to say I liken it to cancer, but there's so many different types of cancer, so not every drug is
Yep.
going to target a cancer. Not every drug is going to target ALS. There's different processes through ALS. So how do you guys, when you do your hits and then when you do your leads, how do you kind of, I guess, navigate through that?
That is a fantastic question. Yes, ALS is not one disease, and there won't be a cure. There will be cures, right? That's what we're striving for. We look at it a couple of different ways. There are therapeutic targets that likely generalize, right? There may be downstream processes that are involved in many types of ALS. But there are also targets that are more specific to specific subtypes. We've seen a great example of this with tofersen, right, or Qalsody. It's a really effective medicine for 2% of people with ALS. So we have to acknowledge that reality, and in some cases, we have programs that are targeted that way. But at the same time, we can't ignore the fact that we don't know those those really early upstream targets for most cases with ALS. So we have to look at some of the generalized targets as well. So we have a balanced portfolio that aims to do both. We wish we could do a lot more of both types. Any other questions? Okay.
All right.
Thank you.
Good.
Thank you.